The last thing we're going to take a look at here is the amino acids and the proteins in terms of the production of proteins or protein synthesis. When we look at amino acids, what we have to remember is that they're going to be large molecules that are going to have an amine group or an amino group within it, the NH2, and a carboxyl group, a COOH, and an R group, and we'll get to what those R groups are here in a second, that will make each amino acid unique. The uniqueness of the amino acids is going to lead to the uniqueness of the proteins based on how those amino acids will bind to each other. And that will give the resultant structure that we look at when we look at the proteins that are there. So what are the different types of amino acids that we might have? The different types of amino acids can be classified based off of the dietary components to it, being essential, non-essential, or conditional. The metabolic action of the amino acids being glucogenic or ketogenic. Or we can take a look at it chemically, which is what we're going to do here, in terms of the types of R groups that we have. In which we have branched chain amino acids. The branched chain amino acids include things like valine, leucine, and isoleucine. Polar, which includes things like serine, threonine, cysteine, tyrosine, asparagine, and glutamine. Polar amino acids such as glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. Acidic amino acids such as aspartic acid and glutamic acid. Basic such as lysine, arginine, and histidine. We also have amino acids that will be neutral. So those are the amino acids that we look at. The way in which the amino acids are going to function is in the production of larger macromolecules known as proteins. And so the proteins are going to come from chains of amino acids, in which we go through a stepwise production of the proteins. The stepwise production of proteins is sometimes referred to as the genetic machinery of the cell, in which we're going to take the codes within the DNA and we're going to translate those codes and transcribe those codes into RNA. The RNA is then going to be able to exit the nucleus, head out and interact with ribosomes, undergo translation, where we're going to change the RNAs, or the nucleic acid codes, into amino acid codes. That amino acid code is then going to undergo assembly, and the assembly will then lead to the functional protein, in which we have distinct steps. The distinct steps include the initiation, the transferal and propagation, and then the termination. What we have to remember is that this is an excessively ATP-dependent process. There's a lot of ATP that's necessary for this process to take place. The way in which we're going to get these proteins to build is based off of the formation of a special type of covalent bond known as a peptide bond. Peptide bonds are going to form in between the amine and the carboxyl ends of the various amino acids. And what we form is a peptide bond, a bond between the nitrogen within the amine and the carbon within the carboxyl. And what we're able to have is a secondary binding site within those amino acids. Once we get the chain of amino acids put together, the ability to form what's referred to as the functional protein is going to be based off of the chemistry that we saw within those specific R groups. The way in which this takes place is that we're going to start by having our chain of amino acids, the AA groups. The chain of amino acids will then start to link together. As they link together, we'll have distinct types of interactions taking place. Those distinct types of interactions taking place are going to change the orientation of the molecules. The change in orientation of the molecules is going to give us two distinct secondary structures, what are referred to as pleated sheets and alpha helixes. You can think of these as being kind of like the inside of cardboard, the corrugated ridges within the cardboard, or kind of like a curl on somebody's hair in terms of the pleated sheet, the inside of the corrugated cardboard, or the spiral, the alpha helix. Those pleated sheets and those alpha helixes start to interact with each other through electrostatic attractions and hydrogen bonding attractions that take place in between the various structures that we have there, leading into our ribbon structures. And then those ribbon structures will then start interacting with each other, forming our third degree, our tertiary, and our fourth degree, our quaternary structures. That's where we start getting the functional proteins. That's where we start getting the proteins that are going to provide the structure for the cells, or going to produce the enzymes that we're going to need for metabolism to take place. Now, within that folding, we're going to get what's referred to as post-translational modifications. That's the bending, folding, and cutting of the amino acids. Not all amino acids that get put into the protein will make it into the protein at the end of all of the folding and bending that takes place. And so what we start seeing various types of attractions taking place in between the various types of R chains within the amino acids, based off of, are they hydrophobic? Are they hydrophilic? Are they nonpolar? Are they polar? Are we able to form hydrogen bonds? Are we able to form disulfide bonds? Are we able to form ionic attractions? Do we have acids and base interactions? One of the things that will form within this, in terms of the hydrogen bonding within the sulfur containing amino acids, where we don't necessarily form a disulfide bond, but we can form what's referred to as a thiol bond, which is a little bit weaker than the disulfide bond. It's more of a hydrogen attraction between the sulfur ends of the amino acids. Will allow for the distinct coiling that takes place within the alpha helixes, whereas the ionic attraction and the hydrophobic, hydrophilic attractions will tend to lead to the sheeting actions that we see within the pleated sheets. All of these chemical reactions that take place, all of these interactions that take place based off of the formation of the unique protein structure, based off of the attraction of the R groups, is what is going to ultimately lead to the structural shape of the protein. And that structural shape of the protein is going to give us the natural function of that protein. So we have to remember that the structure is going to match the function, and the function is going to match the structure. Now, because of the chemical attractions that's taking place within the molecule, this attraction and repulsion that takes place based off of the R groups is going to be dependent upon the environment, the chemical environment that we find this molecule in, which means that any change in the chemistry of the environment is going to lead to a change in the structural shape of the protein, which is going to alter its function. This is where we have two distinct things that will affect proteins more than the other macromolecules. The two things that will affect proteins more than the other macromolecules: heat and pH. Heat and pH, changes in heat, changes in pH, lead to denaturing, and it's that denaturing that will alter the function of the protein. It's why we have to tightly regulate, tightly control our body temperature and our body's pH. Because if we have any excessive change in pH or any excessive change in heat, we will lead to denaturing of the proteins, in particular the enzymes that are necessary for metabolism to take place. So let's take a quick look at what's going on within the action of the sequence here. So we start with the transcription. Transcription, we're going to go ahead and make that mRNA from our DNA so we can get it out of the nucleus. We have to remember that even though we're going to talk specifically about mRNA, we have different types of RNA. We have messenger RNA, the mRNA, but we're also going to have RNAs like the rRNA or the ribosomal RNA. Not only are we going to have the ribosomal RNA, but we'll also have what's referred to as the transfer RNA. The transfer RNA is what's going to bring the amino acids into the sequence of events as it leads into the translation of the code from the nucleic acid code to the amino acid code. So how do we go about making the proteins from the amino acids? And so this is where we have within the DNA two distinct strands that we're going to look at. We have what's referred as the template strand, and that's the DNA strand that's going to lead to the mRNA. And then we have that coding strand, which is simply going to be the complement of what's going on. And so we're simply going to transcribe the coding strand, changing the timing, the T, for the uracil U, because we do not see T in RNA, and we do not see U in DNA. We see U's in RNA, we see T's in DNA. That's how we can make sure that we're looking at the RNA versus looking at the DNA. So the mRNA that's going to come out from our DNA strands is going to look something like this. And so that's great. But what are we going to be able to do with this code? In order to figure out what the code means, we have to have a code breaker. And we have this wonderful kind of template that we can follow to break the code. And so in order to break this code, I have to figure out where am I going to start, where am I going to stop. And so I have this code breaker in which I have my first letter, my second letter, and my third letter. So we have to remember is that we're always going to be looking for 3-letter blocks. And the 3-letter blocks that we first worry about is AUG. AUG is methionine, and methionine is the green light here. It's going to tell me, okay, time to start. And I'm going to go ahead and read in 3-letter blocks all the way through until I get to either UAA, UAG, or UGA, which is going to tell me time to stop. No more amino acids in this chain. And so we're back to our strand of mRNA. And so now I need to look for AUG. After I found AUG, I'm now going to block it off into 3-letter chunks. Those 3-letter chunks are the codons. And so I now need to find my stop codon, either my UAA, UAG, or UGA. Given what I have from AUG to UAA, I'm now going to take that code, and I'm now going to take that sequence from AUG to UAA, and I'm going to start to assemble my amino acids. And so my first letter, my second letter, and my third letter. I start with AUG, then I got CGC, then I got UAA, back to CGC, to CCC, back to CGC, up to UCA, over to CCG, and now I got UAA, stop. So what is this now going to look like in terms of the amino acids? And so if I look at it in terms of the amino acids, I'm going to have methionine, arginine, leucine, arginine, proline, arginine, serine, proline, and then I have a stop. And what's going to happen is that each one of those amino acids are going to start to bond together, and they're going to bond together forming peptide bonds. And following the end of that peptide bonding, they will then undergo their folding, forming the functional protein.